Today, I am going to discuss about the Mendelian genetics. Okay, this one is the second lecture for the part of the series of genetics, and I am Vivek Pattani. Moving forward to the lecture, who is Mendel? Okay, his full name is basically Gregor Johann Mendel. Okay, he was the responsible for the laws governing the inheritance of traits. An Austrian monk who studied the inheritance of traits in the pea plants and developed the laws of inheritance. And his work was not recognized until the start of the 20th century. This is an actual site of the Gregor Mendel's experimental garden in the Czech Republic. Between the year 1856 and 1863, Mendel cultivated and tested some of the 28,000 pea plants. He found that the plant offspring retained the traits of the parents. And Mendel stated that the physical traits are inherited as particles, and Mendel did not know that the particles were actually chromosomes and DNA. Moving forward, these are some of the basic terms. First one is a dominant one. Okay, what do you mean by dominant? The stronger of the two genes expressed in the hybrid is represented by a capital letter. Okay, the one gene which is expressed in a hybrid is a dominant gene, and recessive gene that shows up less often in a cross and is usually represented by small alphabet. And now the genotype. What do you mean genotype? Genotype means a gene combination of a trait. Examples. These are some examples of different types of genotypes. Now phenotypes. Any physical feature resulting from a genotype. For example, various colors of the plants. And now, before moving forward for the Mendelian genetics, okay, let me explain Punnett Square. Okay, let me explain Punnett Square. Okay, what is Punnett Square? Punnett Square is a square diagram that is used to predict the genotypes of a particular cross or a breeding experiment. It is named after the scientist Reginald C. Punnett, who devised this approach, and the diagram is used by the various biologists to determine the probability of an offspring having a particular genotype. Okay, this one here, the diagram shown here is basically an example of a Punnett square. Okay, is example of a Punnett square. Moving forward. The Punnett square shows the complete genotypes of two individuals can produced when they are crossed. Okay, and to draw a square, write all the possible alleles combinations one parent can contribute to its gametes across the top of a box, and all possible alleles combination from the other parent down to the left side, and the alleles combination along the top sides becomes the labels for rows and columns within the squares. The complete genotypes in the square filling it with the alleles from each parent, since all allele combinations are equally likely to occur. And a Punnett square predicts the probability of a cross producing each genotype. I am repeating again, a Punnett square predicts the probability of a cross producing each genotype. Now Punnett square. Okay, Punnett square. How to make a Punnett square? Okay, for example, I have taken one example. Okay, in which I have considered the height of the parents. One is the short one, tall one, and another is the short one. Step one, make a grid. Okay, let's say grid. This one is a grid. Okay, this one is the grid, a square one. Okay, now place the alleles in the gametes of one parent of the top of the grid and those of the parent of the left side. Okay, these are the parents. Okay, these are the parents. Now fill the grid and combine the parent alleles inside the boxes, and the letter shows the genotype of the offspring. Now combine this one, and we'll give it this type of hybrid combination. Okay, this one will give you a hybrid combination, similar for all the squares. Okay, similarly for all the squares. And in step 4, use the law of dominance to determine the phenotypes and phenotype ratio of the offspring. From this one, we can conclude that the genotype ratio is 1 for the all heterozygous, as all of them are heterozygous, and phenotypic ratio is 1 and all are tall plants. Okay, all are tall plants, because the here one parent, okay, possesses a pure tall plant, and as it's a dominant one, it will express in the hybrid form. Okay. Now, Mendelian experiments. Genetics began with the work of Gregor Mendel and developed the basic principles of heredity with no knowledge of genes or chromosomes. Okay, and he did the experiments with pea plants. And why did he choose peas? Okay, first one, variation. Okay, first one is variation. The organisms which are to be chosen for the genetic experiment should have a number of detectable differences at a time. The only single detectable character should be considered. Okay, I am repeating again. The first point is of variation. The organisms which are to be chosen for the genetic experiment should have a number of detectable differences, and at a time only single detectable character should be considered. And the second one is the reproduction. The chosen organism should be sexually reproducing, that is by the fusion of male and female sex cells, because only then offsprings will be able to receive different characters from both male and female parents. Okay, I am repeating again. Why reproduction character is a must? The chosen organism should be sexually reproducing, that is by the fusion of male and female sex cells, because only then the offsprings will able to receive the different characters from both male and female parents. Now, third one of the most important characteristics is the controlled mating. The chosen organism should be able to mate in a controlled or wild plant conditions, because in the genetic experiments, sometimes we have to rear a genetically pure line of parents by methods of controlled mating, and one should be also careful with the records of the offsprings of the various generations. And next point, fourth one, is the short life cycle. Okay, why short life cycle is important? Should have a very short. Okay, it reduces the time of the research. Second one, next one, the large number of offspring. The organism should be able to produce a large number of offsprings also. Okay, how does it help in this case peas plant? Because it helps us to observe variety of seeds, okay, and variety of other characters also, and convenience in handling. Okay, with whatever material we are going to use should be easily be able to handle. The experimental species should be a type that can be raised and maintained conveniently and inexpensively in the laboratory. And as you all know, pea plants are inexpensive, reproduction is easy to control, and produces many offsprings, and also have a contrasting characters, Now, another points, can be grown in a small area. Okay, again, produce lots of offsprings, produce pure plants when allowed to self-pollinate. Okay, produce pure plants when allowed to self-pollinate several generations and can be artificially be cross-pollinated also. Okay, now Mendel's principles. First one is the dominance. Okay, first one is the dominance. One form of the hereditary traits dominates or prevents the expression of the recessive trait. Okay, let's take an example. If in a gene, okay, which represents a character of tall, and another gene which resembles to a character of short one. Okay, and let's take an example. Let's say the tall one is the dominant one. Okay, and these two different are when crossed together. These two are when they both are crossed together will give rise to a hybrid. Okay, and as we know, the tall one is a dominant one. Only the dominant one will be able to express in the phenotype. And the next one is the segregation. And next one is the segregation. Splitting of chromosome during meiosis. Mendel concluded, okay, he concluded that the splitting of chromosome occurs, okay, means separation of chromosomes, okay, and also independently, now next one, recombination, combining chromosomes from both the sperms and egg, okay, combining of chromosomes from both sperm and the egg. Now next one is independent assortment. The independent segregation of genes during the formation of gametes. Okay, meaning independent separation of genes during the formation of gametes. Now Mendel's experimental method. How did he perform experiments? Mendel used hand pollinated flowers using a paintbrush. Okay, given in this figure. Okay, he could snip the stamens. To prevent the self-pollination, and covered each flower with a cloth bag. Okay, and covered each flower with a cloth bag. He traced the traits through the several generations also. And Mendel, okay, selected about 14 true breeding pea plant varieties as a pairs which were similar except for one character with the contrasting traits, and some of the contrasting traits selected were smooth or wrinkled, yellow or green seeds, smooth or inflated pods, green or yellow pods, tall or dwarf plants, etc., etc. Okay, and the Mendel produced pure strains by allowing the plant to self-pollinate for the several generations. Okay, this one is the normal life cycle of a plant in the case of self-pollination. Okay, you all must have studied in the standard 12 also. Here male enter, okay, self-pollination, second fertilization occurs, and germination occurs, and after germination finally the development of the plant occurs. Moving forward, factors. Okay, the term the Mendel used for genes previously was factors. Based on the observations, Mendel proposed that something was being stably passed down and unchanged from the parent to the offsprings through the gametes over successive generations, which he called factors. Now we call the factors as genes, and therefore are the units of inheritance, and they contain the information that is required to express a particular trait in an organism. Now, pair of alleles, one is the homozygous one, meaning the pure one, and heterozygous, which are basically the hybrid ones. Okay, as you all know, the gene which code for a pair of contrasting traits are known as alleles, and they are slightly different forms of the same genes. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at F1 stage and small alphabet for the other trait. For example, in the case of a character of a height, the capital T alphabet is used for the tall trait and the small t alphabet for the dwarf one. And the both capital and the small t's are alleles of each other. Hence, in plant, the pairs of alleles for height would be similar to this one. Okay, this one homozygous, heterozygous, again homozygous. Okay, now genotype and the phenotype. Okay, the Mendel proposed that in a true breeding plant, tall and a dwarf pea, a variety of allelic pair of genes for height are identical or homozygous. Okay, such as both capital T's and both the scenarios of a small t's. Okay, that is one is the dominant one, another is the recessive one. Now the genotype, the genes present in the DNA organism, we will use a pair of letters, okay, to represent them as I previously described, and to represent the genotype for a particular trait. And these two letters in the genotype, because the one code traits from the maternal organism, another comes from the paternal organisms, so every offspring gets two particular codes. Okay, now this one, I'm again repeating it. Genotype, gene present in an organism, usually abbreviated as two letters. Okay. Now this one is a homozygous. This one is an also homozygous. These two, both of them are pure ones, and this one is an heterozygous, which is an hybrid one. Now laws of dominance. Okay. The some alleles are dominant while the others are the recessive ones. An organism with at least one dominant allele will display the effect of the dominant allele one. And during the gamete formation, the alleles for each gene segregate from each other so that each gamete carries only one allele for the each gene. Okay. Now law of independent assortment. The genes for the different traits can segregate independently during the formation of the gametes. Mendel, on the basis of the results of his experiments, recognized the phenomena, okay, below given phenomena. Okay. Now moving forward. Now, it was during the mid-19th century that a headway was made understanding the inheritance. Gregor Mendel conducted a hybridization experiment of the garden peas for about 7 years and proposed the laws of inheritance in the living organisms. Inheritance is the process by which the characters are passed from a parent to the progeny, and it is the basis of the heredity. And variation is a degree by which the progeny differ from their parents. During the Mendel investigations into inheritance patterns, it was the first time that the statistical analysis and the mathematical logic were applied to the problems in the biology. And his experiment had a large sampling size which gave a credibility to the data which he collected, and also the confirmation of his inferences from experiments on the successive generations of these test plants, and proved that his results pointed to the general rules of inheritance rather than unsustained ideas. And Mendel investigated characters in the garden peas plant that were manifested as two opposing traits. Examples are tall and dwarf ones, okay, or yellow or green seeds, which allowed him to set up a basic framework of rules governing the inheritance, which was expanded by the later scientists to account for the all diverse natural observations and complexity inherent in them. And what led to this? Mendel basically selected 14 two breeding plant varieties as a pair which were similar except for one character with the contrasting traits. Okay, now moving forward. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, smooth or inflated pods, green or yellow pods, tall or dwarf plants. Okay, these are the examples of the contrasting traits used by the Mendel. Seed shape round, wrinkled. Seed color yellow, green. Pod shape full constricted. Pod color green, yellow. Pod color green and yellow. Flower color purple, white, flower position axial, terminal, stem, height, tall and dwarf. Now these are the basic ratios of dominant to the recessive ones which he bred. Now in the first scenario, the seed shape, okay, of which round was the dominant one and the recessive one was the wrinkled one. Okay. Now in the F1 generation, all were round. Okay, all were round. The phenotypes were round. In the F2 generation, okay, 5,474 were round and 1,850 were wrinkled, which gave to a ratio of 2.96:1 for the F2 generation. Now seed color, dominant is the yellow one, recessive is the green one. In F1 generation, all were yellow, as yellow is the dominant one. And the F2 generation, 6,022 were yellow, okay, and 2001 were green, which giving rise to a ratio of 3.01:1. And similar in the case of pod shape and pod color, which have had similar ratio, similar to 3:1. Okay. Now the flower color, same, dominant is the purple one, recessive is the white one. And F1 generation, as the purple is the dominant one, all the plants were flower color were purple and ratio was 3.15:1. And the flower position axial dominant one, and the terminal is the recessive one. F1 generation all are axial ones, and F2 generation, okay, 651 were axial and 207 were terminal, okay, giving a ratio of 3.14:1. And finally, from the above results, okay, for totally in the F1 generation all were dominant, okay, in F2 generation 14,949 were dominant and 5,010 were recessive ones, okay, giving a ratio of about 3:1, okay, in the second generation. Now Mendel carried out self-fertilization or cross-fertilization experiment with his bees. In this section, basically examine how he studied inheritance by cross variants with each other. A cross in which a single experimenter observes one character is called a single factor cross, also known as monohybrid cross. Okay, also known as monohybrid cross. A cross between parents with different variants of a same given character produces single character hybrids and also known as monohybrids. Okay, and this experimental approach led the Mendel to propose the law of segregation, segregation, and he collected the seed produced as a result of his cross and grew them to generate plants of first hybrid generation, okay, also known as filial one progeny or F1 generation, and observed that the all F1 progeny were tall and like one of its parents and none were dwarf. And Mendel's analysis, okay, as given in the figure. For each one of the 7 characters, Mendel crossed 2 different true breeding strains. Keep in mind that each cross involved 2 plants that differed in regard to only one of the 7 characters studied. The given example, okay, shown here, basically one cross is between tall and a dwarf plant, okay, and there is a parental cross also. Okay, now this one, a tall plant which is a dominant one, this one is a short plant which is an recessive one. Now all the generations were hybrid and all the plants phenotypes were tall in the F1 generation. Now this one is the Punnett square. Okay, this one is the Punnett square. Now by using Punnett square we can also confirm that all were hybrids and all are tall plants. Moving forward for the first F2 generation. Okay, now what did Mendel do? Mendel crossed the parent plants from the F1 generation, okay, from the F1 generation, and which gave to rise to similar this type of generations in the F2. Okay, similar to this type of generations in the F2, of which produced tall plants, okay, and also dwarf plants, okay, of which some of them are hybrid also, okay, of which some of them are hybrid, some of them are pure also. Okay, now by using the Punnett square, okay, now this one, okay, now this one, okay, which is an pure, will give rise to a pure genotype, which is an tall one and also the dominant one. Okay, now crossing this one, and hybrid variety, okay, again the phenotype will be tall. Again same phenotype will be tall, but this one is a homozygous, short one, okay, homozygous short plant. Allow now interpreting the data, basically the F1 plant of the genotype, okay, when the self-pollinated produces gametes of genotype capital T and small t in equal proportions. When the fertilization takes place, the pollen grains of genotype capital T have a 50% chance to pollinate eggs of genotype T, as well as same scenario for the genotype small t, also the dwarf one, and also the pollen grains of genotype T have 50% chance of pollinating eggs of genotype T, okay, and as well as vice versa. As a result of the random fertilization, the resultant zygotes can be capital pure ones, okay, which are tall, and short ones, and the hybrid ones. And from Punnett square, it is easily seen that the 1/4 of the random fertilization led to tall plants, okay, half one led to tall plants and 1/4 to the TT plants. Okay, so F1 progeny all were tall, okay, so F1 progeny all were tall, okay, at F2 3/4 of plants of were tall, okay, in the F2 3/4 of the plants were tall, where some of the were also pure ones also, and which led to a phenotypic ratio of 3:1, but the genotypic ratio was different, which was 1:2:1, meaning 1 for the tall one, okay, 1 meaning the pure one, 2 for the heterozygous one, and 1 again for the shorter genotype one, okay, which is a recessive one. Now next one, the law of dominance. Based on his observations on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called as principles or laws of inheritance. The first law, or the law of dominance, and second law of segregation. Mendels experiment described that the characters are controlled by discrete units called factors. Factors occurs in pairs, and it is dissimilar, in a dissimilar pair of factors where one member of the pair dominates and another is the recessive one. The law of dominance is used to explain the expression of one of the parental characters in a monohybrid cross of F1 generation and the expression of both in the F2 generation. It also explains how proportion of 3:1 is obtained in the scenario of the phenotype. And next one is the law of segregation. And this law is based on the fact that the alleles do not show any blending, that both the characters are recovered as such in the F2 generation. So one of these is not seen in the F1 stage. So the parents contain two alleles during the gamete formation. The factors or alleles of a pair of segregate from each other such that the gamete receives only one of the two factors, and of course a homozygous parent produces all the gametes that are similar, while the heterozygous one produces two kinds of gametes having each one allele with the equal proportion. Okay, Mendel's 3:1 ratio is consistent also with the law of segregation. Okay, according to the law of segregation, one of the two gene copies present in an organism is distributed to each gamete. It makes an allocation of gene copies random, giving rise to a again phenotypic ratio of 3:1, but a genotypic ratio of 1:2:1. Okay. Now repeating again, the two copies of the gene segregate from each other during the transmission from parent to the offspring. Now, Testcross. Okay, Testcross was first introduced by the Gregor Mendel. Okay, he made the use of Testcross to identify the genotypes responsible for the phenotypes of the progenies after a cross. This means that a Testcross is made between an organism exhibiting a dominant trait, okay, and the another organism displaying the recessive trait. If the recessive trait is manifested in any one of the progenies, it means the unknown genotype is heterozygous dominant. In contrast, if the dominant trait is observed in all progenies, the unknown genotype is the homozygous dominant one. The accuracy of the Testcross results will therefore depend on the availability of large statistical sample. And the crossing an organism with a dominant genotype to a recessive homozygote for a specific phenotype in order to determine the dominance and recessiveness of the unknown genotype and the pattern of the inheritance is purely based on the phenotype of each progeny. Okay, these are the examples of a Testcross. Okay. Going forward, Backcross. What is Backcross? This method has long been used in the horticulture, example to incorporate desirable traits to elite line of crops. At present, the deliberate backcrossing is carried out to breed and produce gene knockouts in the genetic research. To cross a hybrid that is offspring in the F1 generation with the parent one. The offspring produced from such crosses and the process of crossing a hybrid with its parent or another individual with the genotype similar to its parent. Now, backcross, this one, okay, F1 generation, this one is the from parent generation, same is the scenario, this one, okay. Backcross involving dominant parent, backcross involving recessive one, okay, dominant, recessive. Now, as you all know, as this one is the dominant one, all will be tall one. But when we cross between this in this scenario with a recessive plant, one will be tall and another will be dwarf one. Now sometimes there are two different versions of a gene and neither is dominant or the other. A common example which is found in snapdragon flowers, okay. And there are two versions of a gene for flower color. One codes for the red flowers, other for the white flowers. When the plant inherits each copy, okay, of each is a heterozygous, the color is mixed and the resulting flowers are pink. The mixing characteristics is known as incomplete dominance because the neither allele is dominant over the another, and we can't simply use the upper and lower case letters also. Okay, when the experiments on peas were repeated using the other traits in other plants, it was found that the F1 had a phenotype that did not resemble either of the parents, which was in between two. Okay, inheritance of flower color in the dog flower snapdragon is a good example to study to understand incomplete dominance. Now this one, okay, red one, okay, this one is the white flower one. When we cross together in the F1 generation, okay, a pink color give rise to a pink color, okay, give rise to a pink color. Now in the F2 generation, we may check all type of colors, okay. Again, the phenotypic ratio here will be a different one, okay, as compared to the earlier crosses which have been made, okay, in the case of pure dominance ones. Okay, the phenotypic ratio will be of 1:2:1. Okay, again I've been repeating it. The phenotypic ratio will be 1:2:1. Now moving forward for the codominance, when you think of both the alleles, a dominant one, here characteristics do not mix. Rather, heterozygous will display both the characters. Okay, will display both the characters. Using flowers as an example, heterozygous display both red and white patches. Okay, now in the previous scenario of incomplete dominance, there was a mixture of color. Okay, mixture of red and white gave rise to a pink one. But in this scenario, there will be no mixture. Both the colors will be displayed as in the form of patches. Okay, in some flower varieties, we find it possible for two different versions of a gene that code for a different characteristics to both be expressed independently. Again, the phenotypic ratio in this one will be also 1:2:1. I am again repeating it. Phenotypic ratio again will be 1:2:1. Now, let's look at a concrete example of the law of independent assortment. Now, the Mendel's law of independent assortment states that the alleles of two or more different genes sorted into a gametes independently of one another. In other words, allele, a gamete, receives for one gene does not influence the allele received from the another gene. I am again repeating it. An allele, a gamete, receives for one gene does not influence the allele received for the another one. Imagine that we cross two breeding plants, with one with yellow and round seeds, okay, one with yellow and round seeds and one with green and wrinkled seeds. Because each parent is a homozygous one, the law of segregation tells us that the gametes made by wrinkled green plants are RY and the gametes made by round yellow plant all are RY. And in the F1 progeny, similar to this one, where it will be a hybrid ones, Okay, in this case, yellow and round seeds are the dominant ones. Now, question is for you. Okay, you can practice at your time. How many gametes will be produced for the following allele arrangement? And simply remember this formula. Okay, simply remember this formula 2 power n, where n is the number of heterozygous. Okay, where n is the number of heterozygous. Okay, for example, you can also practice for other also. I will be discussing one example here, where in this scenario, okay, how many are heterozygous? 2, 1 and the 2. Total how many? Okay, total 4 gametes. 1, 2, 3, 4. Okay, understood everyone? 1, 2. Okay, 2 to power 2 meaning 4. Okay, now in this scenario, how many heterozygous? 1, 2 and 3. Okay, 1, 2 and 3. Okay, means 2 to power 3 means total number of gametes are 8. Same is the scenario in this example also, where there are 6 heterozygous. Now, according to the linkage hypothesis, okay, the hypothesis, the two genes always stay associated with each other. In contrast, independent assortment hypothesis proposes that the two different genes randomly segregate into the haploid cells. Okay, now I have taken an example of round, okay, wrinkled ones. Okay, 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 there is a mistake. Round and wrinkled, okay, yellow and green one, yellow and green one. Now let's take an example of linked assortment. Okay, let's take an example of linked assortment. Okay, haploid gametes. Okay, these both are haploid gametes. What will be F1 generation? Okay, this will be a hybrid one. Okay, now as per linked assortment, okay, as per linked genes always stay associated with each other, will give rise to a, okay, half will be the round yellow ones, okay, and half one will be green wrinkled ones. Okay, now in the case of independent assortment, okay, now in the case of independent assortment, the first generation remains the same, but there will be the change. Okay, there will be change. Now what happens? Now you can see there will be independent assortment, means 1/4 for the each different types of gametes. 1/4 chances for each type of gametes. Okay. Now Mendel analysis of two-factor crosses. To illustrate Mendel's work, we will consider an experiment in which one of the characters was seed shape and found in round and wrinkled variants. The second character was seed color, which existed as yellow and green variants. Okay, now moving forward for the dicross. Okay, now dihybrid cross. Round yellow seed, wrinkled green seed. Okay, what happens in the F1 generation? Okay, F1 generation. Okay, these are the parent. This one is the F1. This one is the F1 generation. Now what happens due to cross fertilization? Due to cross fertilization in F1 generation, okay, the seeds which will be generated will be round, which will be round yellow one because they are heterozygous. Because they are heterozygous. Okay. Now from the F2 generation, we have this type of arrangement. We have this type of generation. Okay, when we further cross F1 generation with each other, we will give rise to a similar this type of scenario, which is an ratio of phenotypic ratio of 9:3:3:1, of which 9 are yellow round ones. Okay, this one is the green round ones, this one is the yellow wrinkled ones, and this one is the wrinkled green one. Okay, now moving forward, parent plants. Okay, now the Punnett Square. Okay, we will have in the F1 generation in which all are round yellow ones. We have round yellow ones. Now in the F2 generation, okay, by using the Punnett Square again, okay, by using the Punnett Square, okay, by crossing the F1 generation, RY, okay, meaning round yellow. Okay, you can check here carefully. This one, okay, they all are the yellow ones, meaning 9 of them are yellow. Okay, 3 of them are green. Okay, these are the wrinkled yellow ones. These are the wrinkled yellow ones, and one is the wrinkled green one. One is the green one. In this two-factor cross, we need to make a Punnett Square. Okay, this one is the Punnett Square. The phenotype of the resulting offspring are predicted to occur in a ratio of 9:3:1. Okay, now interpreting the data, Mendel results from the two-factor crosses rejected the linkage hypothesis of assortment. Instead, supported the hypothesis that the different characters assort themselves independent. In addition to seeds that were like those of the parental generations, the F2 generation also had a seed that were round and green, and the seeds that were wrinkled and yellow. The two categories of F2 seeds are called non-parentals because these combinations of traits were not found in true breeding plants of the parent generation. The two different genes will randomly assort their alleles during the formation of the haploid cells. Again, I am concluding the two different genes will randomly assort their allies during the formation of a haploid cells. Now the data basically which is of Mendel, the parental one, okay, in F1 generation, as I previously described, all of them are round yellow. In the F2 generation, okay, this one is F2 generation, in which 9, 3, 3 and 1. Okay, we will have a ratio of similar to this one, 9:3:3:1. Now possibilities, okay, a more explanative manner. Okay, these are the gametes. These four are the gametes. Okay, these are the four possible gametes. Okay, these are the four possible gametes. Now this one you can check here carefully. 9 seeds, which are round ones. 3 seeds are round green ones, and 3 will be wrinkled yellow seeds, and rest 1 will be a wrinkled green one. 1 will be a wrinkled green one. Okay. Now, what is the reason behind the independent assortment? The reason for the independent assortment, as we can see in the experiment, we need to fast forward half of the century and discover the genes were physically located on the chromosomes. To be exact, the 2 copies of a gene carried out by an organism, such as Y and a Y allele, okay, are located in the same spot of the 2 chromosomes of a homologous pair. And the homologous chromosomes are similar but not identical. An organism gets one member of the pair from each of its 2 parents. And the physical basis for the law of independent assortment lies in the meiosis 1 of the gamete formation. When homologous pairs line in the random orientations at the middle of the cell, as they prepare to separate, we can get gametes with the different combinations of Mendel homologs and thus alleles on those homologs, because the orientation of each pair is a random one. Okay. Now in meiosis, if meiosis happens many times as it does in a pea plants, we will get both the type of arrangements. Okay, we will get both the type of arrangement. Now in here the metaphase 1, okay, by the chromosome which is a random one, okay, will give rise to a similar this type of gametes. Okay, same scenario in this one also, which is an example of a recessive one. Now second arrangement, okay, again a random one, okay, in the case of an hybrid crosses. Okay. Now finally there are, however, some gene pairs that do not assort independently. When genes are close together on a chromosome, these alleles on the same chromosome tend to be inherited as a unit more frequently than other. And such genes do not display independent assortment, which are said to be linked ones. Okay, there are some examples, okay, which do not follow independent assortment. Okay, they are basically termed as the linked arrangements. Okay, linked. Okay, now finally I am ending my lecture. Okay, thank you everyone. Okay, please subscribe and like the video. Okay, thank you.